The pine, redwood, and other conifers have an ordered growth pattern with an emphasis on the vertical structure. In these trees, the trunk or main stem grows at an accelerated rate when compared to the lateral branches. The vertical orderliness displays strong apical dominance. Unlike conifers, most deciduous trees do not display strong apical dominance. The growth rate of many shoots are equal. This results in numerous branches, which makes it difficult to determine a main stem. Experiments in the 1920s and 1930s by Kenneth Thigpen and Folke Skog determined that auxin was the primary plant hormone involved in apical dominance due to its ability to inhibit the growth of lateral buds. At the shoot tip, auxin is synthesized and loaded into the polar auxin transport stream and is transferred in the basipedal direction towards the axillary bud. This animation illustrates auxin transport down the stem. It enters the cell on the apical end and exits through the pin protein on the basal end of the cell. Here we see the effects of the internal pH of the cell on auxin as it becomes an anionic form and moves down the cell. The external pH is 5, causing the free hydrogen proton to bind with the auxin anion, allowing it to permeate back into the cell. Here, back in the pH of 7, the two dissociate and move towards the hydrogen pump and the pin protein. Viticulture student Robert Lauer explains apical dominance.
The apical bud is at the top where auxin is concentrated to induce growth and suppress the growth of any lateral buds that are here below. When the apical bud would be killed through topping or any other sort of pruning regime, the lateral buds are competing for the new apical dominance. Pruning is an art form and involves miniature landscaping around, say, a dwarf plant. In this case, it would be a vinifera vine. Same idea would go for a bonsai tree. Such a response is the basis for all pruning to form the plant. By pruning carefully, the branching pattern and form of the plant can be controlled, and you can focus on aesthetically pleasing plants, or in the case of vine, would provide ideal grape growing conditions that a winemaker would want from the fruit.
Unfortunately, the mechanisms of apical dominance and the interactions of auxin are still not fully understood. There are many continuing studies in the field, and it is hoped full understanding of the biochemical pathways will soon be within our grasp.